Hello, I'm Arpita Banerjee and welcome to my YouTube channel. For those of you who have been here before, you will find that this video is quite different from the ones that I've posted earlier. In those videos, I discussed theory very little, only wherever necessary. However, in this video, I'm going to discuss a lot of theory, particularly on gene expression and how most proteins could undergo modification for the proteins to become functional. Many of the illustration slides and pictures used in this video have been sourced from the World Wide Web, books, and even lectures that I have attended. You can find the source of the pictures mentioned on the slides in small fonts. At the end of the video, you will find some real research examples derived from the theories discussed. In those parts, the pictures and illustrations come from my own publications or preprints. They have not been taken from anywhere else. So let's begin by talking about what a cell is and how genes get expressed within cells to keep organisms such as us running. We all know that cell is the basic unit of life. Cells make up tissues. Tissues make up organs, and then you get an organism. But the basic unit is cell, and cell's machinery is run by proteins. So the expression of genes that result into proteins is quite crucial. And when we speak of expression of gene, central dogma of life comes into the picture. Cells contain genetic material DNA, which is transcribed into mRNA or messenger RNA, and mRNA is translated into polypeptide that eventually forms proteins. So central dogma of life is about the expression of genes into proteins. On the left, we have an eukaryote cell, and on the right, we have a prokaryote cell. An example of an eukaryote cell is human cell, and an example of a prokaryote cell is bacterium, the plural of which is bacteria. The basic difference is that in a eukaryote, on the left, as you can see, the genetic material is enclosed within nucleus, and the transcription of DNA into mRNA happens within the nucleus. Whereas in case of a prokaryote, the process of transcription doesn't happen within the boundary of a nuclear membrane. Translation occurs in cytosol in either case. There are many factors involved in the process, but broadly, we saw three molecules: DNA, mRNA, and protein. What are their basic units? The double-stranded DNA is comprised of deoxyribonucleotides, which are adenine, guanine, cytosine, and thymine, coded as AGCT. And the single-stranded mRNA is comprised of ribonucleotides, adenine, guanine, cytosine, and uracil, coded as AGCU. A protein, on the other hand, is comprised of amino acids, such as methionine, isoleucine, and serine that you see over here. Three nucleotides in a mRNA code together for an amino acid. The coding is very specific. Just about any three random nucleotides cannot code for an amino acid. For example, AUG is the start codon that codes for methionine, which marks the beginning or the start of a protein. However, any subsequent methionine added later into the polypeptide would have a different code. Therefore, genetic code is very important. However, one should bear in mind that there are variations in the genetic code from one organism to the other. And even there are variations in the mitochondrial genetic code and the nuclear genetic code within the same organism. There are software for translating your nucleotide sequence. Say, for example, in the translate tool of XPASI maintained by Swiss Institute of Bioinformatics, you can paste a nucleotide sequence, DNA or RNA, in the empty box that you see over here, and you will get the translated protein. The best part is that there are genetic code options to choose from. So if you know where your nucleotide sequence is coming from, if you know the organism, if you know whether it is coming from mitochondria or nucleus, then you choose accordingly. That way, the predicted translation is more accurate. Otherwise, you can go for the standard genetic code. So we get mRNA from DNA and protein from mRNA. Now let's dive into their molecular details. DNA is double-stranded and is composed of deoxyribonucleotides. Essentially, it has three components: a nitrogenous base, a sugar residue that you see over here, phosphate groups. These are the three components which make up deoxyribonucleotide. RNA, which is single-stranded, as you see, has a very similar composition, but it is made of ribonucleotides. So if you follow my cursor, you can see the ribonucleotide here. What's the difference between these nucleotides, the ribonucleotide and the deoxyribonucleotide? If you notice this position, the second position carbon, then you will see that in case of ribonucleotide, you have this hydroxyl group over here. Okay, so this oxygen is there. Whereas in case of deoxyribonucleotide, you don't have that. The oxygen is not there. The hydroxyl group is not there. That is the reason it is called deoxyribonucleotide. RNA has also another major difference from DNA. The nitrogenous base thymine is replaced by uracil. The backbone of DNA or RNA is formed by joining together of the nucleotide units. The threading of nucleotides happen when the phosphate of one nucleotide reacts with C3-hydroxyl of the pentose sugar from another nucleotide, releasing a water molecule in the process. Now, how do you get the DNA double strand? That happens by base pairing between two DNA strands. Adenine of one strand forms two hydrogen bonds with thymine of the other strand, and guanine of one strand forms three hydrogen bonds with cytosine of the other strand. The two DNA strands coil around each other to form a double helical structure, where the bases remain on the inside of the helix and the sugar residue and the phosphate groups remain on outside. Now coming to the units that make up proteins. Amino acids are the building blocks of proteins. They come in various shapes and sizes. They have different structures and different polar characters. Some of them are non-polar and some could be charged. The sequence of amino acids, that is, the way they are arranged one after the other, determine the structure of the protein. Now here you see two amino acids coming together and bonding by eliminating a water molecule. Now if you look closely at one of the amino acids, you would see there's amino group on the left, carboxyl group on the right, R side group at the bottom, and the hydrogen on top. All these four groups are connected to the alpha carbon shown in red. In case you're wondering what the R group is, these are the deeply shaded groups that you see in the amino acid chart here. R side group differentiates one type of amino acid from the other. The carboxyl group of one amino acid reacts with the amino group of another to form a peptide bond linkage between the two amino acids. This is how amino acids are joined with each other. The entire process of connecting amino acids to form a polypeptide chain happens within the translation machinery on ribosome. The process is of course finite. There's a stop codon in the open reading frame which tells the process to stop so that you do not get an infinite polypeptide. The polypeptide does not remain as a linear chain. It folds into a functional protein for activity. Therefore, protein folding is dependent on the amino acid sequence and on the composition of the polypeptide chain. That is, what type of amino acids are present and in what order they are present determine the structure and function of the protein. The way a protein folds or the structure of the protein determines its activity. So the protein function is dependent on the protein structure, and the protein structure is dependent on the protein sequence. And there you have a sequence structure function correlation. Now coming back to the central dogma of life, DNA is transcribed into mRNA and mRNA is translated into polypeptide chain and you ultimately get a protein. We require the protein for its activity, right? Because proteins run the cell's machinery. There are various levels of controls that are involved in the entire process that you see over here. Firstly, access to the genetic material or access to the DNA itself is controlled. And then the process of transcription has various controls. Translation too has controls. And last but not the least, there could be chemical modifications that could happen to the polypeptide chain in the cell. And those modifications could be crucial in altering the activity of the protein. Therefore, those modifications could control the protein activity. So there are various levels of controls that could be exercised on the steps involved in the production of a protein in question, as per cellular requirement. If the cell requires more of the protein in question, then its production is ramped up. Also, if that particular protein is not required by the cell at some point of time, then the corresponding gene expression could be suppressed. Now, why do we need such controls? That is, why do we need to control protein activity? Think of an organism which is composed of various organs. Just think of humans. The liver tissues are different from those that are in the heart. The cellular activity of liver cells are different from that of the heart. Hence, the cohort of proteins required for the two organs would differ. So all proteins are not required everywhere at the same time, which is why their activity is controlled as per cellular requirement. Also, depending on the cell's condition at a particular time point, depending on the signal it receives from outside, the cellular requirement concerning protein activity may vary. An apoptotic cell or a dying cell's requirement would differ from that of a dividing cell. Here again, protein activity or gene expression needs to be controlled per cellular requirement. When we speak of controlling protein activity or gene expression, one should bear in mind, however, that all the steps are not simultaneously controlled. So gene expression and/or protein activity is tightly regulated within the cell, and we will soon see how. First, access to the genetic material or DNA within the nucleus is controlled. The nuclear membrane presents itself as a physical barrier, thereby controlling access to the genetic material. And also, tight packaging of DNA ensures that access to the genetic material is regulated. C. Eukaryotic cells present in higher organisms have an additional level of control, the nuclear membrane, which is a physical barrier guarding the genetic material. We know that the presence of nuclear membrane in an eukaryotic cell is a feature that sets it apart from a prokaryotic cell. Now you know why. Nuclear membrane or nuclear envelope acts as a barrier that keeps chromosomes in and the organelles out. The RNA transcribed from eukaryotic DNA usually needs to be processed before it can function properly in the cytoplasm. The nuclear membrane protects the immature RNA from being acted upon by cytoplasmic enzymes. The presence of the nuclear membrane therefore means that the transcription machinery would require transport to and from the nucleus. And there comes into the picture the nuclear pore complex that guard the nuclear pores, thereby serving as gateway in and out of the nucleus. The nuclear pore complex contain proteins that assist in the active transport of transcriptional machinery across the nuclear membrane. And this sort of active transport is required because transcriptional machinery in itself is a huge collection of macromolecules. Transport of such huge assembly of molecules across the membrane requires energy, and therefore the transport is active transport. Now, the first step in the process of transportation of macromolecules in and out of the nucleus is that the proteins require a ticket, import or export ticket. The import ticket is specifically known as nuclear localization signal, which is a specific sequence of amino acids that target the proteins to the nucleus. So the proteins requiring access into the nucleus have such localization signals. So it is clear that not all molecules can cross the nuclear membrane. Most importantly, it exercises control over which and when genes are going to be transcribed. Many transcription factors that would aid in transcription of genes inside the nucleus, such as nuclear factor kappa B or NF-kappa B, have nuclear localization signal. RNA polymerase II that facilitates the process of transcription is a huge multiprotein complex that has other associated proteins attached to it. So the import of such a huge macromolecular complex would require active transport into the nucleus. So in this case, IWR1 protein attaches itself to RNA polymerase II, and this IWR1 protein has the nuclear localization signal. One example of a nuclear localization signal is shown here: proline, proline, lysine, 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 arginine, lysine, valine. Frequency of positively charged residue lysine is quite high here. This specific example is that of a T antigen. Transport out of the nucleus often require nuclear export signal, and one such specific signal sequence example is leucine, alanine, leucine, lysine, leucine, alanine, glycine, leucine, aspartic acid, and isoleucine. So if you notice, it is a leucine-rich sequence. Now, access into the nucleus is controlled, and therefore, transcription factors requiring access to the genetic material is also controlled. And so, masking of nuclear localization signal forms another level of control for gene expression. And such masking has been observed in nuclear factor kappa B. NF kappa B is typically present and resides in cytoplasm in complex with I kappa B or inhibitor family of proteins. Both the size of this complex and I kappa B masking of the nuclear localization sequence of NF kappa B prevents the entry of NF kappa B transcription factor into the nucleus. Upon receiving appropriate stimuli or signal such as cytokines, oxidant-free radicals, ultraviolet irradiation, and bacterial or viral products, and after subsequent downstream signaling, the masking by I kappa B is lost. And exposure of nuclear localization signal of NF kappa B results into its transport into the nucleus, thereby triggering the transcription of genes specially related to inflammatory response. So if you notice, the NF kappa B is an example where the access to the genetic material and the process of transcription initiation are controlled together. Now I will talk about the second point, packaging of DNA that controls access to the genetic material. Packaging of DNA pertains to chromatin structure modification, and we will see how covalent modification of the packaging protein histones leads to chromatin structure modifications. Now, what is chromatin? Family of histone proteins wind up the DNA to form condensed structures known as chromatin. The yellow discs you see are the histone proteins, and the red thread is the DNA. DNA wraps around the histones, and this gives rise to condensed and compact chromatin structures that fit into the nucleus in the form of chromosomes. Not to mention that there are various levels of chromatin structure. Eukaryotic genome is packaged into chromatin structures, and therefore it is inaccessible to transcription machinery. Even before it goes to the level of DNA, the chromatin structure actually undergoes modification to become more accessible to the transcription apparatus. Covalent modification of the packaging protein histones leads to chromatin structure modifications. Now, how does chromatin structure gets modified? Lysine is often present in the N-terminal tails of histone proteins. And lysine can undergo this modification, that is acetylation, where the hydrogen atom of the terminal amino group is replaced by an acetyl group. Now, upon acetylation of N-terminal lysine residues in histones, the chromatin starts unraveling and therefore becomes accessible to transcription machinery. Essentially, the gene is turned on, or the gene gets expressed because of the open conformation adopted by the chromatin. Because of this modification, acetylation of lysine residues in the histone tails. Now, how does this work? How does acetylation of lysine residues in histone proteins unravel the chromatin? Let's get into some molecular details for that. Let me remind you that the DNA backbone has phosphate group, negatively charged phosphate group, and the lysine residues in the histone tails could be positively charged. As DNA wraps around histones, the net negative charge of DNA gets neutralized by N-terminal tail regions of histone proteins. So the attraction between positive and negative makes the wrapping of DNA around histones possible. What happens when the lysine residues in the histone tails get acetylated? The acetyl group by itself has only slightly negative charge, but acetylation results in neutral amide, removing the positive charge of lysine. That would neutralize the positive charge of the histone tails. And the histones can then no longer wind up the negatively charged DNA as it was doing before. With DNA not wrapping itself around histone proteins and with repulsion between the backbone phosphate groups of DNA, the chromatin unwinds. With open conformation of chromatin, the corresponding gene is rendered active for transcription. At the DNA level, class of enzymes called helicases move directionally along a nucleic acid phosphodiester backbone, separating the two hybridized nucleic acid strands, resulting in replication bubble or transcription bubble per requirement. And that is because for the replication machinery or for the transcription machinery to gain access to the genetic material, the DNA has to unwind temporarily to make space for the bulky replication or transcription machinery, because, as I mentioned earlier, these machineries are composed of multiple proteins and hence they require the DNA to open up. Helicases unwind a double-stranded DNA molecule by breaking the hydrogen bonds between the complementary base pairs, allowing the DNA strands to separate. This creates a replication fork which serves as a template for synthesizing new DNA strands or allows for transcription to proceed. Speaking of controlling gene expression through chromatin structure modification, we saw how unwinding of chromatin via acetylation of histone proteins can turn genes on for transcription. Now, how about reversing the situation? Condensing chromatin structure and turning genes off is also regulating gene expression, right? Turning genes off is achieved via methylation of CG-rich regions in the DNA. In this chromatin structure that you see here, the histone proteins are represented by the brown knots or balls, and the DNA is represented by the black string that is wrapping around the histone proteins. The open circles you see in the DNA represent unmethylated cytosins, and under this condition, the chromatin is an open conformation. The dark filled circles you see in the DNA represent methylated cytosins, and under such condition, the chromatin remains in condensed state, thereby turning off transcription. Hypermethylation of DNA essentially results in silencing of genes. Now, such methylation doesn't happen just about anywhere. There are CG-rich regions or CG islands, also known as CPG islands, where frequency of CG, that is cytosine and guanine, occurring next to each other is higher than other regions. These islands are often located around promoter regions in the DNA. In case you are wondering what a promoter region is, the promoter region of a gene engages with RNA polymerase II and transcription factors to initiate the process of transcription. Promoter could span multiple stretches. The region, proximal promoter or core promoter, is just ahead of the coding region, and there could be distal promoter or enhancer regions as well. CPG islands are located around proximal promoter or core promoter region. Now notice below in the picture, with hypermethylated CPG island, transcription is abolished. Here 1, 2, 3 represent exons, and the promoter region is hypermethylated. That blocks its engagement with RNA pol2 and transcription factors, thereby silencing the gene. So CGIs are equipped to influence local chromatin structure, and therefore they can regulate gene expression. Such regulation is achieved via hypermethylation that suppresses the transcription of genes. Now the question is, how does methylated CGIs influence local chromatin structure? Here's my insight. With hypermethylation, all the spots that could possibly undergo methylation get occupied by methyl groups. This results in increased hydrophobic interaction between the adjacent methyl groups. See the blue arrows that is pointing to such interactions? Remember, hydrophobic groups or molecules tend to clump together, so the attraction between the hydrophobic methyl groups would tend to draw the chromatin structure together, resulting in increased winding up of DNA or closed conformation of chromatin. Now, what will happen in such situation? Transcription will turn off, right? In an interestingly coordinated process, proteins that bind to methylated DNA also form complexes with the proteins involved in deacetylation of histones. Therefore, when DNA is methylated, nearby histones are deacetylated, resulting in compounded inhibitory effects on transcription. Likewise, demethylated DNA does not attract deacetylation enzymes to the histones, allowing them to remain acetylated and more mobile, thus promoting transcription. So there's a balance which is maintained. Either you have the open chromatin structure and the gene is turned on, or you have the closed chromatin structure and the gene is turned off. You can't have both, and that makes sense, right? To know further on the controls of gene transcription, such as enhancer elements, silencer elements, to know more about the transcription machinery as a whole, to know more about the translation process and the controls on translation, you can refer to any standard textbook. I read this book, Textbook of Biotechnology by H.K. Das. This book is actually very ancient, like about 2 decades old. I bought this book when I was a master student. This is not to promote any book. You can read any standard textbook of your choice. Let's move on to the protein modification part and see how proteins get modified that alters their activity. Sometimes that modification renders them functional. Now I'm going to talk about how the polypeptide could be modified to control the protein activity. Often it is the modified protein which is functional. So the polypeptide undergoes post-translational modification to be a functional protein. Now we shall see how the finished polypeptide is chemically modified to control protein activity. The modifications are covalent in nature and can take place either during or after assembly of a protein on ribosome. Amino acid residues may be removed or added, which is rare, or the chemical nature of a side chain may be altered. So essentially we are talking about 2 types of modification. One, a chemical group could be linked covalently to the protein via amino acid side chain. This is a list of side chain modifications that could range from phosphorylation to glycosylation to methylation and so on. And the exact residue that undergoes modification is mentioned as well. Here's an illustrative example where you see how the hydroxyl group in the side chain of serine residue could undergo phosphorylation. Threonine and tyrosine residues having hydroxyl group are also capable of undergoing such modification. Just about any serine, threonine or tyrosine does not undergo phosphorylation. So it actually depends on the motif where these residues are placed. So what you see here is the sequence logo of a phosphorylation site motif where serine, the big green S, gets phosphorylated, and the flanking residues that you see have the chances of being present around the serine. In this particular example of phosphorylation motif, the bigger the size of the alphabet, the higher is the chance of that residue being present at that position. 2. Residues could be added or removed. Removal of residues is more common, and in number 2 you see how a protein backbone is cleaved to remove residues. Here hydrolysis of peptide bond is taking place. We will soon see real examples of these 2 types of modifications. Now how do these modifications affect protein activity? So the effect of post-translational modifications could range from conformation change to localization change to chemical property change, and these effects are seen on the protein. This is the sequence alignment of the cysteine protease proteins in Necator americanus, which is hookworm. And now I'll show you some regions and illustrate localization change. This alignment comes from a piece of work deposited in bioRxiv. The title of the work is Cysteine Proteases of Human Hookworm, Necator americanus, as Virulence Factors and Implications for Drug Design with Antiheparin and Heparin Analogs, a bioinformatics study. So this is the piece of work, and on the right hand there's a link for downloading the PDF file. This is the first page of the PDF where the author contributions are clearly mentioned. I did this piece of work in collaboration with some scientists at University of California, San Diego. I had worked there from March 2016 to December 2017 for about 2 years, and I finished collecting data for 2 projects in that time period. 2 projects. I am not presenting the entire papers here, okay? Only some excerpts which are relevant to the topic being discussed here, that is gene expression and protein modification. Now, let's get back to the topic which was being discussed. Cysteine proteases. Cysteine proteases are essentially protein degrading enzymes, that is, they're capable of destroying other proteins. These enzymes have cysteines in their active site. This bioarchive preprint documents the molecular characterization that I did of the 7 cysteine proteases found in human hookworm. Coming back to the sequence alignment to demonstrate how post-translational modification could be associated with localization change. This is a slightly zoomed in version of the alignment. Look at the extreme upper left. You would see a black bar on top demarcating the secretory signal. If you follow the start and the end point of the bar in the alignment, you would get the predicted secretory signal, which directs the cysteine proteases 1, 2, 3, 4, 4b, 5, 6 to the outside. CP7 doesn't have it, and so it doesn't get secreted. Notice the blank space in the alignment. This corresponds to the CP7 sequence of human hookworm. Now, this diagram is again from the scientific article I mentioned earlier. Notice on the extreme left that CP7 remains inside the pathogen, whereas the rest of the cysteine proteases are secreted out into the host extracellular matrix. What do you think these proteins do over there? Remember, I told you that cysteine proteases are enzymes that are capable of destroying other proteins. Well, these enzymes of hookworm degrade host proteins. That is, they degrade human proteins, which is why they are secreted out of the pathogen. What happens after the proteins are secreted out? The signal sequence that directed them outwards get removed. And that happens by cleaving of the peptide bond where the signal ends. So we are talking here about a post-translational modification, that is removal of sequence, involved in localization of the protein. The secreted proteins remain outside after the signal sequence is removed. Though localization precedes the modification in this case, after the modification, the cysteine proteases remain outside the pathogen. Now, what happens after the proteins are localized? Let's take a look at the alignment in figure 1. Again, zooming in, take a look at the proline in the alignment, which is marked by a blue box. Notice the P's boxed in blue. This is the start position of the mature polypeptides. Okay, and on further right, you see the cysteine residues that are boxed within red. That's the active cysteine residue of the cysteine proteases. That is, these cysteine residues are within the enzymatic cleft and they participate in the enzyme reaction. If you look closely, you would see that CP7 has a D instead of a cysteine. That is, an aspartic acid instead of a cysteine. So CP7 is not enzymatically active. Also, as I mentioned earlier, CP7 lacks the secretory signal. It's not even secreted out. The motif that you see boxed within purple in CP7 was actually predicted to be a lysosomal targeting pattern. Notice that there are a stretch of amino acid residues between the start point of mature polypeptide and the end point of the secretory signal, right? So even after the proteins get localized to their destination, that is, after the secretory signal gets removed, there's this stretch of peptide that keeps the secreted proteins in immature form known as zymosens. The proteins remain in inactive form in this state. Now take a look at these three-dimensional structures of CP1 to CP6. All of these proteins have the active site cysteine residue and they're enzymatically active only after the propeptide is removed. You see that cleft over here in the protein, a very clear cleft, that's the active site. And the propeptide, when it is present, it blocks this active site. Therefore, the stretch of amino acid residues between the start point of mature polypeptide and The endpoint of the secretory signal, shown by the red bar here, gets enzymatically cleaved off, which results in the conformation change of the zymosins, and then they turn into active cysteine proteases capable of degrading other proteins. So here's another example where cleavage of protein backbone is involved in converting immature zymosins into active protease structures. Now I'm going to talk about an example of covalent modification, where the side chain of an amino acid of the protein gets modified, and that could result in chemical property change. In this particular example, I'll talk about covalent binding of heparin to the cysteine proteases of hookworm. Heparins are glycosaminoglycans that include molecules such as hyaluronan, chondroitin sulfate, heparin sulfate. In this particular image, what you see is the chondroitin sulfate attached to the core protein via serine. The linkage to serine is covalent in nature. In case of hookworm cysteine proteases, the covalent linkage of heparin is via asparagine residue. In figure 1 of the mentioned manuscript, you will find a region demarcated as N-glycosylation motif region. This is where the heparin molecules are predicted to bind. This is zoomed in version of the alignment, and if you see at the N-glycosylation motif region, at the start position, you can find the asparagine or the N residue in most of the cysteine proteases. This is where heparin molecules gets covalently linked to the enzymes. When the cysteine proteases leave the pathogen, when they're secreted from the pathogen, they already have heparin molecules, at least most of them have heparin molecules bound to them. The heparin molecules are indigenous to the worm. What purpose do they serve? Why are those heparin required in the first place? Heparin in bound form acts as a cofactor to the enzymes and possibly exercises allosteric control over the enzymatic action, making the NACPs functional even at alkaline pH, which is required for the survival of hookworm that encounters different pH conditions in its life cycle within the host. I further postulated that upon release of heparin molecules from the cysteine proteases by deglycosylation enzymes present in the small intestine of human host, heparin possibly serves as a cofactor to antithrombin, which inhibits thrombin by binding to it, thereby interfering with the coagulation cascade, leading to blood thinning of the host. When hookworm sucks blood from a human host, it would not want the host blood to get coagulated. Coagulation would mean interference in blood sucking, and therefore the pathogen actually targets the coagulation cascade of its human host. Protein phosphorylation is one of the most important modifications that lead to a diverse array of biological response, such as metabolism, hormone action, cell growth, and therefore cancer, gene expression, memory, etc. As mentioned earlier, the residues that primarily get modified are serine, threonine, and tyrosine because of the hydroxyl group they have. Enzymes that carry out phosphorylation of proteins are known as protein kinases, and the enzymes that carry out the reaction in the reverse direction, that is, enzymes that dephosphorylate proteins, are known as phosphatases. Now, for the purpose of showing you a real example, this is the profile alignment of N protein or nucleocapsid protein of Chandipura virus isolates with other representative rhabdoviruses. Alignment comes from one of my publications, Whole Genomes of Chandipura virus isolates and Comparative Analysis with Other Rhabdoviruses. It's an open access paper. This is the first page of the PDF of the article, and you can see that I and Rashmi S. Gunjikar contributed equally to the work. Rashmi was actually part of the sequencing team, whereas I had done the bioinformatics analysis of the sequences. Speaking of rhabdoviruses, you will see rabies virus in the alignment. This Chandipura virus work had been done by me at National Institute of Virology, Pune. I had worked there from October 2007 to April 2010. For about 2.5 years, I got 2 publications from the work I did at the institute, and I'll be presenting relevant parts from one of my publications. Going into the paper, you will find the profile alignment, which I'm going to talk about a little. This profile alignment of N protein has sequences of viruses belonging to family Rhabdoviridae, genus Vesiculovirus, and genus Lisavirus. Profile alignment mode of Clustal X was used to create this alignment, where profile 1 was made up of sequences consisting of genus Vesiculovirus, that includes Chandipura virus, and profile 2 was made up of sequences of genus Lisavirus, that includes rabies virus. If you notice in the phylogenetic tree, you'd find that the vesiculoviruses are distantly related with the Lisa viruses. The purpose of the profile alignment was to glean information from other well-characterized viruses that would not otherwise align very well with the comparatively newer and somewhat distantly related Chandipura virus. Here, in this part of the alignment shown within the blue oval are the putative CAM phosphorylation sites in the end proteins of the viruses. The motif residues are in bold font, and if you notice the motif in case of CIN6514, it begins with a K or lysine, whereas the other representative Chandipura viral isolates CIN0327 and CIN0728 have a R or arginine. Though K and R are somewhat similar in the context that both are positively charged, but nonetheless, they are different amino acids with different properties. Now, why does it matter? See, CIN6514 isolate comes from the first reported Chandipura viral outbreak in Maharashtra that happened in 1965. Back then, the virus was not causing encephalitis in children, whereas the other isolates are from outbreaks from other geographical parts in the years 2003 and 2007. And these were causing encephalitis. So the virus had definitely evolved to be much deadlier. And it goes without saying that most changes happening at the gene level translates to the protein level and has the potential to contribute to final biological outcome. In this context, it is imperative to mention that the mutation is appearing within the N-terminal region containing the first 47 amino acid residues, which have been shown to be critical for self-assembly of nucleocapsid protein. Now, there's another such example in this part of the alignment. If you look within the pink oval, you would see the predicted CK2 phosphorylation sites in some viruses here. Upon closer inspection, you'd find that a motif for CIN6514 or the isolate from the 1965 outbreak is SRED, and the motif for other Chandipura isolates are SRDD. So we have a E or glutamic acid which is getting replaced by D or aspartic acid as the virus has evolved. Again, though E and D are somewhat similar in the context that both could become negatively charged, but nonetheless, they are different amino acids with different properties. In this context, it is imperative to mention that the deadlier Chandipura viruses are accumulating the mutation at a place which in case of Lisa viruses has been shown to be critical for viral RNA transcription and replication. Also, deriving from the alignment, the region could be delineated as an important T-cell epitope. More importantly, the mutation appears in a region which has been shown to be important for n-RNA interaction in Chandipura viruses. Now, in both the cases I mentioned, the exact residue which undergoes modification is not changing. Speaking of the CAM phosphorylation site here, if you look at the Chandipura viral isolates, then threonine is the residue which could undergo phosphorylation. But that remains same. What changes is some other residue in the motif. Now, turning our attention to the CK2 phosphorylation site, if you look at the Chandipura viral isolates, S or serine is the residue which could undergo phosphorylation. That remains the same. But what changes is some other residue in the motif. So how does it matter? See, the binding of kinases or phosphorylating enzymes depend on the flanking residues or the motif residues as well. So even seemingly insignificant or small changes, such as a change from K to R or from E to D, has the potential to impact the level of phosphorylation of N proteins and hence their functioning, making the proteins from the deadliest strains different from the prototype. ScanProsite is my go-to tool for motif prediction, as ProSite is a database of protein domains, families, and functional sites. You can search ProSite by motif IDs or by sheer name of the motif. You can even scan a protein of your interest for predicting putative motif. I'm really thankful to Sarah ma'am who gave me an opportunity to work in her lab at National Institute of Virology. And I'm also thankful to Vidya ma'am who provided me funding for work. Most of the theories and the real research examples that I presented here had been compiled by me way back in July 2019 when I was teaching these to a PhD student at Indian Statistical Institute, Kolkata. I was volunteering there. I hope you find the content useful and I will make the next video when I can. Thank you very much.